My name's Steve Webb. I'm the CEO of the Global Institute for Food Security, and welcome to today's webinar, Exploring Advancements in Plant Science and Accelerated Plant Breeding, or breeding, because as we learned at our last seminar, it's not just plants, it's also plants and animals. And I'm joining you from our office in Saskatoon, Saskatchewan, Canada. And as we gather here today, we acknowledge that we are on Treaty 6 territory and homeland of the Métis. We pay our respects to the First Nation and Métis ancestors of this place and reaffirm our relationship with one another. Thank you to everyone for joining us today. At GIFS, we take pride in being a connector and an innovation catalyst within the agri-food sector, and events like this are one of the ways we can do just that. Today's seminar is the second event we're hosting, Exploring Accelerated Breeding, which describes a suite of technologies that combined help optimize breeding pipelines and to accelerate the rate of genetic gain, whether they're for plants or animals. And the end result is bringing impactful improvements in breeding material, whether they're plants or animals, to producers faster. I know many of you did attend our last seminar, the first in the series in July, and our intention for this event is to pick up where that one left off, furthering our discussions and taking a deeper dive into the genomic selection and predictive breeding aspects of it. If you joined us in July, you were fortunate to hear from Dr. Herman Spangenberg, a professor emeritus at La Trobe University and former head of Agriculture Victoria Research. And Herman is a member of GIFS International Science Advisory Panel. At the event, Herman provided a great overview of some of the successful applications of the technology, its starting point in dairy, and then its expansion across many crops. And he's been very instrumental in helping us pull together today's seminar. And I want to thank him for his continued support of the Global Institute for Food Security. This is truly an international event. The presenters and attendees are joining us from around the world, here in Canada all the way to Australia, which is already tomorrow today. Depending on where you are, it might be late or early, and I appreciate you taking time to fit these into your busy schedule, and we do encourage discussion. We'll do a Q&A session at the end of the presentations today, but please enter any questions into the Q&A section on the Zoom screen. And of course, if you have any technical issues, reach out to the moderator and we'll help address those as we can. To get as many questions as we can today, but we want to be cognizant of everyone's time, and we'll keep the seminar within the 2-hour window we've planned. If there are any questions unanswered, we will do our best to follow up with you after the event and get back to you with an answer. Before we start the program, I want to provide you a little background on the Global Institute for Food Security and the work that we do. Next slide, Dan. And again, GIFS is an interesting partnership between industry, government, and academia, established in 2012. And Nutrien, the government of Saskatchewan, and the University of Saskatchewan are the founding partners. We were created in 2012 to address global food insecurity and the innovation challenge. And we're created to help capitalize on Saskatchewan's unique end-to-end strengths in agriculture and food and biotechnology globally. We serve as a connector and an agri-food catalyst, partnering with groups from government, academia, and industry here in Canada, but also around the world, to help discover, develop, and deliver innovative solutions for the production of globally sustainable food. Next slide. We do this by investing in relevant technology platforms that transform scientific capabilities. Next slide, Dan. Sorry, I didn't tell you that. And again, one of the roles that we play as the connector and catalyst is to advance innovation through our people and access to our capabilities and platforms. We bridge commercialization from lab bench all the way to working with companies that have products in the field. And John just came back from a field tour of a company that's got a product that's in the field, and we serve as a connection bridge. And again, I think is one of the advantages for GIFS. Best example of that is our relationship with Bangladesh, where we recognize the opportunity for partnering with Bangladesh was much bigger than what the Global Institute for Food Security can do on its own. And I know Leah Olson, the former CEO of PAMI, is on the call, and Leah and her team were are terrific partners in that relationship because they bring expertise that complements and helps us advance the mission of global food security around the world, along with the College of Ag Science, Global Institute for Water Security, NRC, and the Food Center. And that's an example of us acting as a connector. Next slide. Our programs, this is a structure. This is Nancy Tout's organization. Nancy's the CSO and she's on vacation, but on the call as well today. Our programs and platforms are designed to be at scale and to be pointed at market opportunities and enhance synergy across our ecosystem, not only within GIFS, but within the ecosystem here in Canada. Our technology platforms support research and development and enhance digital agriculture, accelerated breeding, and increase quality crop yields. Our facilities here in Saskatoon are state-of-the-art, and including our Omics and Precision Agricultural Lab, the first analytical facility of its kind in Canada for combining genomics, phenomics, and bioinformatics services to agriculture, Canada's agriculture and agri-food sectors. OPAL is a significant component of GIFS Accelerated Breeding Program that is being mobilized to serve public and private partners across Canada and abroad. You'll hear a little bit more about that soon. I want to introduce GIFS leader of this area, John Davies, and I'd like to welcome Dr. John Davies. Dr. Davies is a former professor at Iowa State University, a fellow at Dow AgriSciences, now Corteva, who's assisting in the development of GIFS Accelerated Breeding Program. Today, John will discuss why accelerated breeding is needed to help agricultural producers address many of the challenges affecting production and meet growing global demand for food. He'll also discuss the technology and the talent that GIFS has brought to Saskatchewan to help us serve the private and public plant breeders in this province, in Canada, and beyond. Please join me in welcoming John Davies.
Thank you, Steve. Get my slides. Okay. Can you see my slides? Good. Thank you. I'd first like to talk about why some of the challenges in feeding the world. As everybody knows, the population of the world continues to increase. It's increasing about 20% over the next 30 or so years, from about 7.7 billion people today to approximately 9.7 billion in 2050. And we still need to feed people on limited resources of land, water, and nutrients. In addition, we have an additional challenge with climate change having a negative impact on much of the food production throughout the world. We have a challenge as well that conventional breeding timelines are slow, and the current genetic gains are somewhat insufficient to meet our future needs. There's a real need to increase innovation in food production and sustainability. Accelerated breeding is a solution for this. It enhances sustainable food production and increases yield. It enables breeding to changing environments. It can increase the return on investment in breeding programs, and it facilitates breeding of multiple traits simultaneously. Accelerated breeding at its heart is a combination of technologies, but one that's really important is genomic selection. Genomic selection is using phenotypes and genotypes to predict phenotypes in future populations. It's been used in a number of different species, and it's shown to be able to increase genetic gain. On the right of this slide, you're seeing phenotypic selection, which is a traditional way of plant breeding, and it's showing continued genetic gain over time. And this is shown has proven very successful over time. However, it has been shown to be improved if genomic selection is also applied to this, and can be further improved if we do things like speed breeding, which enhances the drive to homozygosity in plant breeding populations. So accelerated breeding is all based on the breeder's equation, which says the genetic gain is equal to the selection intensity, the selection accuracy, and the additive genetic variance over the selection interval, how long it takes to do a round of genetic selection. Accelerated breeding enhances the rate of genetic gain by increasing the selection intensity and the accuracy and decreasing the selection interval. And it's based on technologies such as genomic selection, speed breeding or double haploid formation, high throughput phenotyping, as well as breeding optimization. If you were here with us last month, you heard Herman Spangenberg give a very nice presentation highlighting aspects of accelerated breeding. What's shown here is a number of different species in which genomic selection has been applied. One that I'd like to highlight is the use of genomic selection in dairy profitability. And here it's showing a balanced performance index over time and the advent of using genomic selection back in around 2000. You can see there was a significant increase in the rate of genetic gain in the balanced performance index. In addition to that, if you look at individual traits, you can see that a number of traits were contributing to this balanced selection index and highlighting that multiple traits can be worked on simultaneously in improving the performance of this. In addition to working in dairy cattle, other species have been used at Agricultural Victoria Research. And what's shown on this slide is the genetic gain in ryegrass, where it was shown to support a 5-fold increase in the rate of genetic gain over just 2 rounds of genomic selection. So significant increases in a relatively short period of time. In addition to ryegrass, new pea varieties have been produced using genomic selection, and that's shown on this slide. In addition, genomic selection has been applied to wheat breeding, and here is showing some work on wheat rests, and in canola, showing genomic selection advancing varieties at Agricultural Victoria Research as well. So as I mentioned, the technologies that are used to enhance genetic gain include speed breeding or double haploid technologies that reduce the time to homozygosity, genomic selection, which rapidly identifies new lines that can serve as parents and reduces the time required for field testing of new lines, high throughput phenotyping, which reduces labor costs and can lead to earlier decisions, and then simulating and optimizing breeding schemes. When you bring in these new technologies, oftentimes the old way of breeding is no longer the best way to do it. And being able to simulate breeding schemes can actually help optimize this. And these technologies increase genetic gain per cycle and decrease the time required for new products. So why accelerated breeding? Again, it shortens the time of the breeding cycle. It enhances the rate of genetic gain through increased selection intensity and accuracy and decreasing the selection interval. And it increases the genetic gain per dollar invested. Increases in cycle time, increased selection pressure using GEBVs, and more optimal use of resources. GIFS has developed a number of technologies to support accelerated breeding, and we're looking for partners to partner with us. Our genotyping is housed in our OCL platform, which has high throughput sequencing and high throughput genotyping. Genomic selection is managed in the data management and analytics group, which can do genomic selection, optimal parent selection, and plot vision for phenotyping. Reduced cycle time is housed in cell biology in our plant growth facilities, with cell biology doing double haploid formation and our plant growth facilities developing speed breeding techniques. In addition, we're modeling to optimize breeding pipelines in our data management and analytics group. So accelerated breeding is GIFS is looking for potential partners for accelerated breeding. This is one format where we can work together with breeding organizations to do that, where the plant breeding partners provide phenotypic data and plant samples, We can then genotype those plant samples and use the phenotypic data along with the genotypic data to develop genomic prediction models. In the line development, if we're provided with parental crosses from F1 seed, we can produce lines either through doubled haploid formation or speed breeding. We'll then genotype those doubled haploid or speed bred lines and grow them to seed. We can use the genotype data along with the genomic prediction model to calculate new GBVs and to select plants, use those to select plants for field testing in future generations. We're very excited about these technologies and are looking forward to working with potential partners on that. Please give us a call when you have a chance. You can contact us at partnerwithus@gifs.ca. Thank you very much. At this time, I'd like to pass it over to our guest from Agricultural Victoria Research. I'd first like to introduce Dr. Michelle Malmberg. Michelle is a senior research scientist in the molecular genetics team with Agricultural Victoria Research. She leads the bioinformatic research and sequence analysis and developing genomic pipelines and their application for approaches such as genomic selection. She specializes in forage, medicinal, and oilseed species with particular focus on ryegrass, alfalfa, and cannabis. Michelle's presentation will explore genomic selection in orphan species. Please join me in welcoming Michelle.
Thank you, John. Can you see that? All right. Thank you. Yes, thank you for inviting me and giving me the chance to come and talk to you today about some of the work that we do in AVR and how we would go about enabling genomic selection in orphan species. Orphan species being those that have little or no genomic resources available. And in the agricultural space, that typically means plants. So you can be talking a lot about plant species throughout this talk. I'm going to take you through three sort of broad areas, starting with how you would start to build up some of those genomic resources, particularly focusing on developing a reference genome. How you then might go about choosing an appropriate genotyping method for genomic selection, and then through to designing a genomic selection approach. Something I'm not going to really talk very much about today, but is very vital for genomic selection is obviously phenotypes. You need both excellent phenotypes and genotypes to implement genomic selection. There's a range of ways you can collect those phenotypes, and the best approach really will depend on your trait of interest. So let's start with building up some genomic resources. The first place you're probably going to want to start is to have a look at if there are any existing resources available at all for the species of interest. So typically, there will at least be some idea of an estimated genome size and diploidy, so how many copies of the genome the species has, but this is not always the case. Reference genomes are typically not available. Sometimes a closely related species will have a reference genome that can be used to some degree of success. You also want to see if there's any sequencing data available, such as on online repositories. Are there any known detected variants? Gene annotation, although if we're an orphan species, that is quite unlikely. Perhaps there are just a set of particular genes of interest that are well known in the species that you might want to make sure to focus on. And something else that can be thought of as a resource are plants themselves. So in your species, are there elite varieties or cultivars available, or are you primarily looking at wild populations? Is there a set of mapping populations that might help you to narrow down the diversity that you're looking at, or even just a commonly used set of reference plants, for instance? And what is available to your species will be very different, really depending on what you're working on. Throughout this whole process, there are a number of species-specific considerations just to keep in mind that will really have an influence on how you approach, particularly genotyping for genomic selection. And I'll go through these one by one. So if we start with genome size, again, as I mentioned, talking a lot about plants. This is a graph just showing the various distributions of genome size that you see in different groups. So in the top near the top red arrow there is flowering plants, and you can see that grey bar covers the whole width of this plot, whereas next to that second red arrow you see mammals, which have a much smaller range of genome sizes present. And another way that we can look at this is to graph it as a bar plot. So on the left there in black, so small that you almost can't see it on this plot, we have some microbial genome sizes. And in the middle in orange, we can see some animals, a bit more variation there, but still a fairly tightly conserved range of genome sizes. And then on the right, you see plants. So we have some quite small genomes again, quite hard to see on this graph, becoming quite large in common species such as wheat. And then on the right, I've also included Parastrapunca, which is the largest known plant genome. And on this graph, it goes up to 35 gig, but it continues up the side and goes all the way up to 150 gig. So quite a range in genome sizes there. With the plants in particular, you're also going to have issues around ploidy. So plants can be diploid or polyploid. And you can also have a mix of ploidy within the same species. So ryegrass has both diploid and polyploid varieties. And if your species is polyploid, that can then be either autopolyploid, where it has multiple copies of the same genome, or allopolyploid, where it has one or two or more genomes combined. So an example that you can see on the right there is Brassicinatus or canola. That's an allopolyploid. It has two subgenomes from two different species, the A and the C genome. And what you can see in there is different colored blocks that represent ancestral karyotypes, which have been duplicated and inverted and spread throughout the genome. So we have quite a lot of genome duplication present, both within the subgenomes and between them. And this is genome duplication is very common, particularly in plants. In the bottom there, you can see a synteny graph of the three subgenomes of wheat, and you can see a lot of similarity between those subgenomes. You also have to think about how your species of interest breeds. This ranges quite drastically in plants. So you have a lot of species that are self-incompatible and they don't cross with each other, such as ryegrass. And then you have, for instance, canola that's able to self quite happily. And then you have things like potato, which although they're outcrossing, you can then clonally propagate. And this is all going to have an effect on how diverse your species is and what your expected level of heterozygosity might be. And I will also just point out that little graph in the corner, in the left-hand corner, is showing that as you increase in ploidy as well, it takes more generations of inbreeding to reach a homozygous state. So there's additional things to consider if you are highly heterozygous and also polyploid. So all these things are sort of factors that need to be kept in the back of your mind as you're going through this whole process. But if you're a genomic orphan species, you're going to probably want to start with assembling a reference genome. This will most likely include a combination of different sequencing approaches, but these days, really the backbone of any reference genome assembly is going to be some form of long-read sequencing, such as using a nanopore backfire. Then might want to do some mapping and linkage using potentially high-C or gamma nano to create scaffolds, and then potentially apply some short-read sequencing to polish those reads and remove errors, and potentially some transcriptome sequencing to help with gene identification. There's a range of software available to do this, and this is by no means an exhaustive list. This is an area that seems to still be evolving with new programs coming out regularly, and what's most appropriate will depend on the type of sequencing that you use and also potentially what species you're working with. But generally, you want to assemble your genome, do some form of scaffolding and polishing. Some genome annotation is a good idea, as well as some QC to make sure that everything is looking sensible and making sense. And something that I think we're moving a bit more to is pan genomes, where you combine information from multiple assemblies, particularly incorporating additional gene information. And you can do this using publicly available genomes as they are released, or you can incorporate in-house genomes that you've assembled yourself. I will also say that if you are not an institute with access to this whole suite of tools that will give you a really nice reference genome, there are also commercially available services for both sequencing and assembly, so that can do the whole process, or you can choose simply a niche service or step as well. And there are lots of options out there. So now I just want to give a bit of an example of a pan-genome reference that is currently being put together by a PhD student with us at Agriculture Victoria Research. This is in ryegrass, and what she's done is taken a publicly available reference genome PIAS, which you can see in that top bar on the graph on the right. PIAS is a haploid assembly of ryegrass, which means that it's a very good level assembly with very little duplication present. And in that cluster chart, you can see that it's all mostly single copy, but there is still quite a significant chunk of expected gene information that's missing. So she's going to add additional information to this core reference genome by incorporating regions of the genome that are present in an additional 6 or 7 in-house assembled genomes that are not present in PIAS, hoping to reduce the proportion of missing data without introducing too much more duplication. And this is going to cover the diversity of both perennial and short-term ryegrass as well. Something similar has been done by another PhD student at AVR for cannabis, taking a publicly available reference and adding 6 de novo genomes to this core reference. And she's done a really nice analysis looking at the predicted genes in this pan-genome. So in total, she found over 43,000 genes, but only 23,000 of those were 4 to all 7 genomes. About 1,900 were unique to single genome, and about 18,000 were present in 2 or more. And it just goes to show that if you are creating a reference assembly based on a single individual, you do miss out on a lot of genomic diversity and particularly gene information. I think this approach is going to become more common, particularly as more reference genomes become publicly released. So that's sort of the first step. You develop up a reference genome, and then the next thing that you might want to do before you launch into genomic section is to do some variant discovery and a little bit of diversity analysis to become more familiar with the species you're working with. Typically, you'll sequence a set of plants that are representative of the diversity that you are interested in using whole genome sequencing. Essentially, this ideally should be as large as you can afford to make this data set, but it should be representative of the material you're interested in working with down the line as well. So for variant discovery, you'll typically primarily look for SNPs, but there are other variants as well that you might want to consider, such as indels, presence absence variants, and copy number variants. We'll do some filtering on that, and I'll touch again on that later. And I do just want to make a note that if you are working with a polyploid species, this step is going to be a little bit more difficult for you, simply because a lot of softwares don't cater to polyploid genotype pooling. There are ways to get around this, and there are programs that do this, but you will potentially end up with a little bit messier data. But that's okay. Genomic selection really is a very robust method and can tolerate some degree of error quite well. So it's still worth pursuing, but it will be a bit of an additional hurdle for you. So once you know where some of the variants are across your genome, you might want to do a bit of a diversity analysis. One easy quick way to do this is to look at a neighbor joining diagram, also known as a diversity tree, to see if there's any population structure. So on the side in the top right, you can see a tree representing cannabis diversity. Bottom right is canola, and the two down the bottom to the left and in the middle are both ryegrass. And the reason I've included 2 ryegrass is to show that the tree on the left includes diversity from both short-term ryegrass and perennial ryegrass. And you can see quite a bit of a structure and branching present there. But in the tree in the middle, that's 50 populations that we were working with in a crossing program at AVR. And you can see that there's a lot less structure present when you just take a smaller subset. You might also want to have a look at and doing a MOVA, look at diversity within versus between your populations. For genomic selection, having an idea of what the linkage disequilibrium is in the population that you're working with is a good idea, because that will inform you on the minimum number of markers that you need to capture diversity across the genome. That's sort of the first step. You've built up some resources, you have a reference, you know where the variants are in your genome, and you have some idea of what the population that you're working with looks like. The next step is to select a genotyping method to apply in genomic selection. So there have been quite a lot of advances in sequencing technology, and they do keep going. That graph on the right shows the cost of genotype sequencing falling really dramatically. And although that graph cuts off, I think in 2014, it's continued to be very true that cost of genotyping The economies of scale that we can achieve these days are quite impressive, and we keep getting better. This is just also to show that there's now really a wide range of different machines and technologies that you can use as well. And again, particularly for this genotyping method, you are going to have to think carefully about your species before selecting a method. For instance, if you have a very large genome size, doing whole genome sequencing is probably going to be prohibitively expensive for you. If you have a polyploid species, you might need to ensure that you have additional sequencing depth to make sure that you are capturing all of the alleles that are present in the species, for instance. And then you also have to consider where are you applying this genotype data. So when we were doing resource development, the sequencing method was probably the most important thing to think about, really focusing on the sequencing. When you're doing diversity analysis, the sample set that you're using is probably the most important to make sure that you're capturing all of the relevant diversity going forward. For genomic selection, you sort of need to think about, am I applying this to a breeding program? And if so, at what step? Am I sequencing the parents, or am I sequencing offspring where maybe I already have some genotype information for the parents? Or am I doing some cultivar or arrival improvement? All this will influence and affect what the most appropriate genotyping method would be. You also have to think about how many samples you need to do and the depth of coverage that you do require. So there's a range of options out there, and there's no one size fits all, and you also are unlikely to simply pick one and only do one method. You can combine multiple sequencing methods within one breeding program using perhaps different methods at different stages. It really depends on how you're applying it. The only one I'm not going to go through in any great detail is long read versus short read sequencing. So long read sequencing currently, due to its high error rate, is typically used for reference genome assembly or to detect very large structural variants, but not yet for routine genotyping. I'll go through the rest in a little bit more detail. So whole genome sequencing is just we sequence the whole genome at depth, and this is a great method. It gives you deep coverage of the whole genome, and it covers both genic and intergenic regions, which can be quite important for genomic selection. It gives you information about promoter regions and enhancer regions and things like that. It's an open genotyping system, so it doesn't require any development. You buy a kit, you make the library, and you sequence it, and it's quite simple. But this method is expensive due to the sequencing cost primarily, and especially if your genome is very large, this method is probably not going to be suitable for you. The method that's become really quite popular is skim whole genome sequencing. It's essentially the same thing. You make the same library, but you just generate less sequencing data and you save some money that way. So it's more cost effective. You're still getting coverage of the whole genome, and it can be imputed accurately if, especially if you have some related samples that have deep sequencing available. Same thing, it's an open genotyping system, no development required, but it does require imputation, and although that can be fairly accurate, it's not 100% perfect. You will have some errors there. It can also be difficult to accurately genotype heterozygous plants, because if you have two alleles but you happen to only get one read, you could miss one. Any sequencing errors that come through are more likely to result in genotyping errors. And if you have a very specific variant of interest that you want to capture, there's no guarantee that it will be. You will have random dropout and missing data throughout the genome due to just the low coverage that you're generating. Nonetheless, this is the method that I really quite like, and there's a lot of programs now have been developed to work specifically with skim data. And so particularly if your genome is a little bit on the smaller side, this is a very attractive approach. There are another couple of methods here that are aimed at reducing the gene space that you need to actually sequence. The first is transcriptome genotyping by sequencing, also known as RNA-seq. This is a method that we had used extensively in the past at ABR, particularly with ryegrass, where you sequence the RNA, so the expressed genes, and this gives you a normalization of the genome size. So the gene space across plants is much more tightly conserved, so you end up sequencing less of the genome. RNA shearing can be a little bit easier than DNA shearing. And again, it's an open system. You buy a kit, you make a library, and you sequence it. But it does use RNA, which is quite unstable and it can be problematic if you are receiving samples from quite far away. Another method that is very popular in plants is RAD-seq. It's also sometimes just known as GBS. And again, it gives you a reduction in the portion of the genome that you actually end up sequencing. but it does have a high proportion of recent data, and it can be particularly unsuitable for outbreeding species because it does have quite a lot of dominant markers. It's not one that we've personally applied at AVR. And then lastly, I have put together genotyping arrays, which are, for instance, SNP-GIFS and GBS target assays, because these are both probe-based methods. So the genotyping arrays do require some specialized equipment to run, whereas the target assays, the GBS target assays, use probes to pull out your areas of interest from the library and you only sequence those. Both these approaches are highly accurate and give you very good coverage of your regions of interest. They have low rates of missing genotypes, and they can be very cost effective at scale. And they also have very well established lab protocols. But they do both require quite a bit of investment to develop. Although that I think is getting a little bit easier these days, particularly with the target assays. Not all variants, however, will be suitable for capture probe design. So you may have a variant that you know that is very important and relevant biologically. However, that doesn't mean that you're going to be able to successfully design a probe to bind to that region. So biology and functionality don't always seem to eye to eye, so that's something to keep in mind there. I'm just going to give you a bit of an example of where we've moved our genotyping and ryegrass at AVR. So we used to do transcriptome sequencing, but there were issues around RNA stability. So now we have moved to a target capture assay where we've designed a set of 27,000 probes that actually covers over 72,000 SNPs because SNPs tend to cluster in the genome. Our lab team has done a lot of work around liquid handle robotics to help with automation and should also reduce the reaction volume required. We've achieved our reduction in off-target sequencing goals. So the majority of our sequencing we get from this is now on our regions of interest. And we validated this in both perennial and short-term ryegrasses and also had a look at population. So this is now a really cost-effective and highly reproducible routine genotyping method for us that we use regularly. And as I said, we also tested this in populations. So sometimes we're not interested in sequencing individuals, but whole cultivars of populations in ryegrass. So we wanted to see if the probe was able to capture the allele frequencies basically across the whole populations. And to do this, we had a look at a set of plants from the cultivar Kidman, where each leaf was harvested individually from each individual and processed as individual libraries. We then took those same plants and harvested them in bulk. We then took a large set of seeds from the same cultivar, harvested them in bulk, and then compared that to a bulk harvest of seeds from a different cultivar. What you can see on the graph on the right is that Rohan sits nice and far removed from the three different versions of Kidman that we sequenced to show that the Kidmans, they're all genetically very similar, sort of capturing the same information whether we harvest leaf individually or if we harvest in bulk from seed. We had a look at how many individuals, though, do you need to include to capture the genetic profile of a cultivar, and we do need at least 40 individuals to truthfully represent that cultivar. And you also need quite a high sequencing depth, so around 200x coverage of your target SNPs is ideal, which is why a target capture approach is so attractive, because it allows us to reduce the portion of the genome we need to sequence, and then we can afford to sequence that to a high depth. So that's sort of where we're at currently, but we plan to improve upon that further and really take some learn from what the bovine world has done, which has advanced quite far in genetic section and how they genotype the genetic section. And one of the ways that they've improved this is to incorporate functional mutations, as they are more likely to be causal. So that's something that we're going to have a look at in the future. We're also thinking about designing a low coverage assay. Say, for instance, you more deeply sequence your parents and then you go to sequence next generation, and you don't need perhaps as much information. You could apply a low coverage assay. And again, this is something that's been done very successfully already in the bovine world. So this is where we're thinking to move to in the future. So those are some of the methods that you can use to sequence, and with the exception of the genotyping assays, the SNP-GIFS, all of these methods involve some form of sequencing data that you're then going to want to process. This is just a rough outline, and again, there's not an exhaustive list of the tools or the programs that you can use, but very generically, you're going to want to do some sort of QC, trim your sequences, align them to your reference genome. It's a good idea to mark PCR duplicates if your library protocol includes PCR. You might want to do some base close for recalibration if that's available to you. You then pull variants, filter those variants, and you have to impute any missing data because genomic selection requires complete data. I'm just going to talk you through those last two steps. So you're going to want to filter to ensure that you have high confidence genotype pools, and depending on what depth of coverage you aimed for with your sequencing, that's going to inform what a suitable minimum depth cutoff and maximum as well. What an appropriate cutoff will be for those. You'll need to decide on how much missing data you're willing to tolerate. The more missing data that you put into imputation, the higher your error rate is going to be. What's a suitable minor allele frequency cutoff, and that will depend on things like how many samples you're working with. And something that you may wish to apply would be something like what's your maximum expected heterozygosity. So for instance, ryegrass is a highly heterozygous species and I would not apply this filter. But when I was working with canola, the samples we were working with were expected to be almost completely homozygous, but we were getting quite a bit of heterozygosity coming through simply because those two subgenomes are very similar. So you had some reads just aligning randomly between very similar regions in those two subgenomes and appearing as heterozygous SNPs. So you might want to filter on that, and more importantly, probably control for alignment, by looking at alignments. Again, just reiterating that you have to approach this keeping your species-specific considerations in mind. And as I mentioned, you have to impute up any missing data because genomic selection doesn't handle missing data. There are a lot of imputation programs out there. Many do require an ordered genome and a reference panel of some sort, but if you are an orphan species, you are unlikely to have those available to you. There are programs that use instead linkage to equilibrium and K nearest neighbor approaches that do not. So link impute is one that we use very successfully with ryegrass like Antibio. Some of the things you have to be aware of though is the effect, as I mentioned, of missing data. So that table on the right in the middle just shows that as your proportion of missing data increases, your imputation accuracy rate falls. It's still pretty good, and as I mentioned, genomic selection is a very robust method. It tolerates even up to 10% error rate quite well. So this can help just to inform how much missing data you're willing to accept and how much error you're willing to put into the work that you're doing. Something else to consider is your minor allele frequency. If you have a low MAP of 0.05 and 95% of your samples will have the same genotype, it's not hard to predict the missing sample has the same genotype. Whereas if you have a slightly higher MAP, your accuracy is going to fall a little bit. And then depending on what sequencing method you decide to go for, if you went with a skim method, it's worth noting that there are some imputation programs that are specifically designed for skim data, and it's definitely worth having a look into them if that's the approach that you decided to go with. And as I mentioned before, you might consider using a combination of sequencing approaches for your genomic selection. So here's an example in ryegrass where we had our parental generation, Gen 0, where we had 100 samples that was sequenced to high coverage. We then had the offspring, the next generation, Gen 1, where we had 517 samples that were initially also sequenced to a high depth, but that we then computationally subset down that sequencing data to simulate skim sequencing at 3 different levels. So at 1.5x, 1x, and 0.5x coverage of the genome. We then imputed these skim samples using the parental high coverage genotypes as well to give us a bit more information. And what we can see on the graphs on the bottom left there is that as your coverage of the genome drops, so does your imputation accuracy, which is what we would expect. But if you actually then take that through genomic selection and you have a look at the GBVs that are generated from either these skimmed down genotypes or versus the GBVs calculated using the original high coverage data that we had available for those samples, you see very good, very high correlation across those 3 skim levels. So our rank is preserved, showing that we can sequence that offspring generation at a much lower coverage, which saves us some costs of sequencing and potentially allows us to reallocate those funds to sequence more individuals, which is beneficial for the selection. That's the first part. You've got a reference genome and some resources, and you've now had a good think and you've selected your genotyping method going forward. You now need to think about how you're going to implement genomic selection. So this is really about how you genotype and phenotype your reference population to develop a prediction equation, and then genotype the selection candidates to then pick your plants to go forward with. And again, as I said, phenotypes, accurate phenotypes are vital. You might want to have a look at things like trait heritability to make sure that the trait of interest can be selected for, have some idea of how complex it is. And knowing trait heritability will also inform you on your maximum expected genomic selection accuracy, so how accurate can you realistically expect it to be. Particularly with plants, you're going to want to incorporate things like seasonal measurements and multiple traits, and to have a look at something like the selection index to combine the weight of those traits against each other. And something I'm really not going to go into much detail on, but also some consideration should be given to what model to use. So there's a range available with different underlying assumptions, which might be better suited to certain traits. You're going to want to have a think about incorporating genotype by environment effects. Inbreeding, mitigating inbreeding is also going to be important, particularly if you're working in an obligate outbreeding species, which can often have quite a load of recessive deleterious mutations present with high inbreeding depression. And that's something to keep in mind. And also, again, with plants, you might want to consider things like a restriction window so that you can group your plants by flowering time or plant breeders' rights traits to make sure that your resulting populations are nice and uniform, for instance. And we also have to think about where are we implementing genomic selection. So if we're doing this as part of the breeding program, at what point of the breeding program are we targeting? Are we picking plants to mate, or are we picking the varieties that even go into the breeding program from the start? And this really will depend on what the current breeding practices are for your species of interest. You can do things like mate pair selection, where you pick individual plants to combine for breeding, for crossing, or you can do something like genomic subsection, which is also sometimes known as genomic speed breeding, where you take an existing cultivar and just keep the top X% of individuals. If you're working with an orphan species, the first place you're probably going to start with is a within population prediction evaluation, because you won't have any data from previous years to inform the current year. But as you then start to build up these resources and to implement genomic selection, you might want to start thinking about a forward prediction approach where you use any previous year's data to predict into the current year. And if you're wanting to do a cross-population prediction, it's really important to have a think about your reference population and make sure that you've captured enough diversity that is going to be relevant across populations. So I'm going to talk you through just a bit of an example in ryegrass, and I wouldn't by any means call ryegrass an orphan species. It definitely has quite a few resources behind it, especially now. But it's a nice example of the evolution of genomic selection implementation over time, and how it's gotten more complex and how it's been built on. So for biomass yield, genetic gain has been quite limited in ryegrass, particularly if you compare it to other global plant species. And there's a number of reasons for this. It's an obligate outbreeding species. It's really high heterogeneity. There's a lot more diversity within a cultivar than between them. And we also get poor correlation between an individual or and a sort in terms of biomass performance. And a breeding program is traditionally quite long, around 10 years. So the place where genomic selection sort of started in ryegrass at AVR was to have a look at 20 years worth of historical data from the breeding company and to simulate the genomic prediction accuracies that could have achieved had they implemented genomic selection throughout their breeding program using a forward prediction approach. So using all of the previous year's data to predict into the current year. And what they found was moderate prediction accuracy was achieved across the years, which for yield is quite good, given it's a very complex trait. And this was initially implemented at the year 5 stage of the breeding program, so taking clonal rows and selecting out sets of 4 plants to cross in new synthetics. And this is a better picture of where genomic selection is in ryegrass today. So we have a lot of custom software and pipelines developed up to help apply genomic selection in the most effective way. We incorporate things like heading date groupings to ensure that they're flowering together. We include a fitness matrix to try and mitigate the effect of inbreeding. We have restriction windows around plants' breeders' right traits to ensure uniformity of the populations that we create. For instance, the point of plant selection has been moved up from that year 5 stage to much earlier in the breeding cycle, up to year 1 or year 2, I think. So reducing the breeding time by a number of years and therefore increasing the genetic gain. Just incorporating a lot of different things together and really building up genomic selection from something simple to now incorporating a number of traits and approaches. So this is a mate pair study selection approach. We're selecting, in this case, 4 individual plants to cross together to make a synthetic. As I mentioned, you can also implement genetic selection using a subselection approach. So particularly if you're working in an orphan species, because not a lot of breeding work has been done to them often, there's typically high diversity and very little differentiation in varieties that may be available. So to take an existing variety and select out the best performing individuals using genomic selection with the checkbook approach, it's something that we've tested in ryegrass and it's worked quite well, particularly as ryegrass cultivars are quite diverse. I will just note that if you have a species with known high inbreeding depression, that's something that does need to be managed to make sure that we end up reducing performance due to too much inbreeding. But this method works well and it's quite rapid and can be cycled through very quickly. So to summarize, I just want to say, keeping in mind at all times your species-specific consideration, how big is the genome, what complexities do you have to deal with in your species, throughout all 3 of those steps, from building up genomic resources, choosing a gene typing method, and then designing a genomic selection approach. And although there are fewer resources, more uncertainty, and potentially more errors in orphan plant species, I again just want to say genomic selection is a robust method. It can tolerate some error and still lead to significant improvement in genetic gain. So it's definitely worth pursuing. Thank you.
Thank you very much, Michelle. That was a great presentation. Once more, we're going to hold our Q&A session to the end of both seminars today. If you do have any questions, please enter them into the Q&A or the chat box at the base of the screen, and we'll follow up with them after Abdul speaks. So our second speaker is Dr. Abdul Jighly. He's the acting research leader for the computation biology team at Agriculture Victoria Research. He has an extensive career spanning over 13 years. He's successfully adapted and harnessed the diverse spectrum of technologies ranging from traditional farming practices to cutting-edge genomics, computational techniques, and digital agriculture. His pioneering efforts around the conception and realization of innovative multidisciplinary statistical solutions that synergize the informativeness of different statistical models and data types. Notably, he's made contributions to extend the inception and refinement of groundbreaking statistical models like CGM, WGP, SNP-MACE, 3GS, and PolySim. And the impact of these models has played a pivotal role in advancing current practices to meet future global food security goals. Today, Abdul's presentation will address genomic selection and sustainable food security. Please join me in welcoming Abdul. Abdul?
Thank you, John. Thanks for the introduction and thank you for the invitation. Let me just first try to share my slides. Okay, so yeah. So yeah, thanks again for the invitation. My talk is continuing on what Michelle started on genotyping and genome prediction, but I will dive more into genomic prediction and what are the future, how we see the future at Agriculture Victoria, how we see the future should be for genomic prediction, what are the models that we are going to apply in the future, and things that go beyond genomic prediction. And my presentation will be in this structure. We'll start with just mentioning the old model that we used to do, like linkage mapping and genome-wide association, and why you are applying genomic selection. and the basics of genomic selections and how we are going to go beyond genomic selection. And we'll give one example, and which is one of the most promising approach by integrating growth model with genomic prediction. So linkage mapping and genome-wide association, they are both traditionally used by plant breeders and also by animal scientists, especially for genome-wide association, and they both aim to detect quantitative trait loci. And linkage mapping, for example, work on families. So we have 2 or more parents. We have crosses between them. We generate F1 and then we go to F2 and so on. Because so our population will have some specific structure, but the whole diversity will come from the number of parents we started with, just with having crosses between them. On the other hand, genome-wide association would exploit what we call historical recombination or historical recombinations between lines. And we start from a diverse population of different unrelated individuals. So the ancestral history of the population will be something like that, multiple colors, while in the QTL mapping, we have some specific ancestral haplotypes or parental lines, which are the ones that we start with and we know about them. And just to compare them on the chromosome level, what's happening, assume that we have here a gene, and in linkage or in linkage mapping, what we have usually is very high LD, and usually it doesn't take us to need a high marker density. So with low marker density, we can work quite well with detecting the genes or with linkage mapping. But the problem will be, because of the limited number of recombinations, we will have a low resolution detecting the genes. So for example, here, this whole area could be potentially the position of the gene. But if we have low marker density for genome-wide association, you can see that the gene cannot be detected because none of the marker is really linked to it. But if we have high marker density, we can detect the gene, and at much higher resolution, so a very small proportion of the chromosome will be, or the confidence interval of the location of the gene will be within a very small proportion of the chromosome. So, and this is the main difference between them. But after that, the thinking come, especially for the purpose of selection, that GWAS or linkage mapping, what they care about is that can detect only the QTL with large effects. And usually for most trait and most complex trait like grain yields, for example, very small proportion of the total genetic variation is really controlled by these major QTLs, and that is not enough to make selection. But in selection, what we really care about is the merit of the individual, regardless of what gene it has. So we just need to give a kind of relative comparison between different genotypes or different individuals to know the best one. We don't really care what genes are in that individual if we want to select for it. So genomic selection, what it does, alternative to GWAS or linkage mapping, is that it estimates the effect of all variants simultaneously. And in this case, what we can get is that it can capture the whole genetic variance explained by the marker without really knowing what genes are involved in that, or without needing to know what are the most effective SNPs. So it just to estimate a kind of what we call the genomic estimated breeding value, which is represent or equivalent to the phenotype, and just to know the merit of the individual. And the beauty of genomic selection models, that they are very flexible. So we can fit genotype by environment interaction. We can use environmental data, for example, or other source of data such as expression or omics, or for example, management data. And it's flexible enough to be integrated with other models, which is one of them I will explain today, which is crop growth model. So just to illustrate how genomic selection works, we start usually with a reference, what we call the reference population, which is usually has phenotype and genotype. And we use this population to train the model or to calculate the prediction equation. And what I mean by prediction equation, that because the reference population has genotype and it has, for example, SNPs or variants, different type of variants, we try to give a weight for each variant, and what we call the SNP effect, for example. We give that effect how much, and this represents how much the variant or the SNP affect the trait. And then we have a different set of population, which we usually call the selection candidates, which has genotype only. And then we apply, and it should be genotype with the same type of SNPs that we calculate the effect for them. And then we use this effect to estimate what we call genomic estimated breeding values. And based on that, we select the best candidates of these selection candidates. So the reference population, you can usually also hear the term training population, which is the same. It should have phenotypic and genotypic records. And we train our genomic selection model on it. And increasing its size usually lead to increase the prediction of accuracy to some extent. And it should be related to the selection candidates. So if you have the ability to make your reference population very related to the selection candidate, that will be much better. And of course, we use it to calculate genomic estimated breeding value for the selection candidates. Selection candidates, again, they should have genotypic records. But usually in research, you see that the selection candidates will have genotype and phenotype, and that just for the purpose of research to estimate the prediction accuracy of the model. And it's not used for training the model. It just should be independent from the set that is used for training the model. And the prediction equation that we calculated from the reference, we applied to them to estimate the genomic estimated breeding values. And there are multiple, if you take to the linear model, the standard linear model, there are different genomic selection models, and you will hear lots of names, different Bayes with another letter, or G-blob or R-blob. They are all, in theory, they are all the same, but they are not just the main difference between them is the distribution of SNP effect, how they assume that SNP effect is distributed. For example, we have the G-blob and Bayes C. They assume that the effect of the SNPs, so if you have genotype your population with 100,000 of SNPs, it assumes that the effect of the SNPs should follow a normal distribution. Bayes C would allow a normal distribution for some SNPs, while the remaining SNP will have a zero effect. So just on the zero. So a subset of the SNPs will have zero effect, and then the remaining will come from their effect will come from normal distribution. Another method like Bayes A and Bayes B will have what we call the t-distribution, which will allow for a little bit higher frequency of larger effect QTL, something here and here. There are also the double exponential distributions such as Bayesian lasso, and which also which allow for a little bit larger proportion of larger SNP effects. And there is also the most comprehensive one, which is Bayes R, which assumes four different a number of distribution. Usually we use four. One with zero. We assume that part of the population have zero effect. We have also some small effect, medium effect, and large effect, QTL or variants. And what the best method? Well, Bayes R is the most comprehensive one, but usually the best method for you is depend on the trait. So which model or which distribution will truly fit the effect of the SNPs will be the best model for that trait. So it's more about the traits, how it's complex or not. So if you know that you have something like disease resistance where you have some major genes and minor genes, so maybe Bayes R will be the best option for that, for a trait like that, because it allows you to have large proportion of large effect SNPs, but still you have lots of zeros and lots of small effects QTL. But for trait like yield, usually the normal distribution with G-blob usually work well. And it's usually, in most cases that I have seen, it's work as equivalent to Bayes R. So they have usually the same accuracy. So it's just how your trait or how the distribution fit your trait, or the effect of SNPs for your traits. So estimating prediction accuracy is one of the most important concepts that when you run a genomic selection study. And with what we do usually to predict the accuracy is that we mask or we hide a proportion of the population of the phenotypes and we assume that we don't have it, and we run our prediction equation, or we calculate our prediction equation on the remaining individuals. And usually what we do is we use the one that we masked. We try to calculate GBVs for the masked individuals and trying to see how much they are correlated with the true phenotypes, or how much the correlation is between the genotypes or the GEBVs and the true phenotypes. And it has different forms, and each one has, of course, applications. So the first one is to completely mask a subset of the individuals. And we usually use this scenario if you want to know the accuracy of predicting new lines. So if you are within breeding program and you develop the next generation of the population of the next generation, and you still don't have phenotypes for them, and you want to select based on GEBVs. So this scenario, if you run this scenario for prediction accuracy, it will mimic that case of having new individuals into the integrate to the population. So you can calculate their GEBVs in this case. Another form is just to randomly mask different plots or different individuals across different environments. And this is why we usually do that. Imagine that you have 10 field trials, and in each field trials, a subset of your population is planted there. So a different subset in different environments, and then you run your model and you try to get an estimation for the whole population in all environments. So just trying to move from something like moving from unbalanced set to a balanced set, where you have phenotypic records for all individuals in all environments. The third time, which is the most tricky, the trickiest one, is to mask a completely one environment and trying to predict it based on its relation with the other environments. And this is actually mimic the case when you want to predict the performance in future climates, which is one of the hardest tasks we have in predictive genetics. You may also hear the term key-fold cross-validation, and in this strategy for cross-validation, what we do is we divide the population into different subsets. For example, five-fold, we call it five-fold prediction. So we divide our population to 5 sets, and in each one of the sets is used for validation and the other for the reference. So we start, for example, from the first one as validation, then the second one as validation, the third one as validation, the fourth, the fifth one as validation, and we repeat this for 10 or 20 times. And this is mainly to estimate the confidence interval of the accuracy. So, for example, we have 5 folds multiplied by 20 times, which means 100. In total, we have 100 replicates, and 100 replicates for the accuracy. We estimate the mean and standard deviation, so we have a better understanding of our accuracy, our prediction accuracy, and how much it's, what is the standard deviation for the accuracy. So this is why we repeat it multiple times. And there are lots of successful examples. It's really beyond being described, and John today described some of them. So for example, on wheat, on canola, on barley and oat, there are different literature that show that, and something is especially important for food security in Africa, something like cassava. So there are also successfully applied for different traits on all these crops. And there are, if you just Google for genomic prediction, you'll find application in almost all major and minor crops that we are using. But actually, applying genome prediction alone is not really enough to achieve future or global food security. So we need to have even more advanced method to really accelerate our genetic gain to the level that we need, or for the future, for example, 2050. So before I explain why we should go beyond genomic selection, I would just love to explain what are the current practices the industry use. So if you look at how different industries, whether in plant or animal, they are applying genomic selection, you will find whether they are using Bayesian, whether they are using linear mixed model like gblob style or whatever, you will find that most of the practical application, fall within one of 3 categories in terms of how they do or which model they use for genomic selection. So the first one is if you have, for example, multiple environments in your reference population, they analyze each one independently and ignore the intersection between environment. And by intersection, I mean that how much genetic variation is shared across different environments. And in this case, because you run each environment independently, the three environments, for example, in this example, the three environments have no relation to each other because you are running each one independently. And just for illustration, you will get an estimation of SNP effect that is representative for the environment itself. So something in the middle of the environment, the SNP effects will be. And there is another type of analysis, but in this case, in the independent case, you will have a SNP effects for each environment. You can fit all the environments together and assume that the environment is as a fixed effect, something very statistical term, but it just kind of getting the average of all environment in simple words. And you get a single SNP effect estimation or single GEBV per individual. But the prediction, what you are really predicting is something here in the intersection of all your environments. So this is the shared genetic variation across all your data set. And this type of, and this type of analysis will give you this. The third type of analysis people are usually practicing today is the genotype by environment interaction, which is the most comprehensive and usually result in the most accurate results. Is that like the independent one, it creates a SNP effect for each environment. But because we are telling the model that we have three different environments and these environments are related to each other in somehow genetically. So instead of having the prediction in the middle of the environment that is descriptive specifically for the environment, you will find that the prediction or the SNP effect will be shifted toward the shared zone across the three environments here. So it's still representative for the environment, but it borrows some information from related environments. So which is usually resulted to the most accurate prediction for the environment itself. And the other thing, not just for what people are using, is how difficult to take change. I will just love always to mention this example, the first genomic selection paper, which was published in 2001 by Mike Goddard, Ben Hayes, and Tim Houston. And if you look here at the citation, the first 7 years, the paper didn't got any citation, even though it's now reached, I think, something around 6,000 citations. This slide is old. So it reached something around 6,000 citations now. But the first 7 years, it didn't really get any citation. And this is mainly because one reason is the technology wasn't there at that time because it was very forward-thinking method at that time. But also adapting change is not really usually easily. So it took for animal, for example, breeding to start to applying it until 2007, and for the plant communities took around 2010. But from 2007 to 10 till now, till today, the application is, as you have seen, the three examples that I presented in the last slide. And even though there are lots of methods has been developed, lots of models, but none of them are really getting applied in practice or rarely getting applied. So let me just now talk about the limitation of genomic selection, why we should go even beyond genomic selection. So genomic selection by itself is not enough anymore. So the first thing is that let's just look at dividing the phenotype as three components, which are the genetic genotype by environment interaction and environmental variation. So there are the three main components for the phenotype, the variation of the phenotype. So with genomic selection, we model G by E. This modeling is really partial. It's not really explaining the actual G by E. And we don't model E. We can sometimes fit E as fixed effect or fitting some environmental covariates, but that's not a modeling of E. That's just trying to remove some of its effect. So it's not included in the GEPVs, but it's just to remove their effect. And for some traits, they have really low heritability, something like 5%, 2%, 3%. We have some commercial traits and important traits that are like that. And for this trait, G by E and E are really important to model them. And some of them also have high G by E. And if you want to, with genomic prediction, usually, if you, or the standard model that I explained, if you want to go to predict a new environment, usually the prediction accuracy is very low. And the same when you're trying to have individuals that are not related to your reference population. If you're just trying to predict a new population that is, if you, in simple word, on PCA, it's just clustered away from the reference population, the prediction will be really low for these individuals. So genome selection, in general, it works on G and G by E usually, and G by E is partial. It's not really complete or comprehensive modeling for that. And the solution for that is to develop novel integrative approach that integrate different models or different data types together. And in this presentation, I'll focus on one example, which is one of the most promising examples, is by integrating genome prediction with crop growth models. What are crop growth models? It's just a kind of deterministic simulation for the plant life. Just from the first day you plant your seed to the day you harvest your materials, a day-by-day simulation for that, and you try to model each processes that happen every day. So, for example, you in this day, was the temperature, this was the environmental input. I have fertilization to what our management. I have fertilization and irrigation at these dates. So the soil has this amount of water, it has this amount of nitrogen, and the seed can take this amount of inputs or environmental inputs every day. So it's something like equivalent to when you go to a nutritional expert and they told you, if you eat this amount of calories every day and you practice this amount of time, you are more likely to lose this amount of kilograms per month. So it's something similar to that. So it's just trying a kind of a biophysical modeling for the process of the plant. And to do that, what we usually use is something called genotype-specific parameters, or it's usually called the latent traits, which is generally a trait with a very, very high heritability. In theory, it's 1. Their heritability is 1. And they are not interacting with the environment, but we know through equation or deterministic modeling or through specific equation how these GSPs are interacting with the environment. So for example, one of things that people may be familiar with is vernalization. So some individual or some plants may be have very low sensitivity to environment and to vernalization, and others have high sensitivity to vernalization. And we know how vernalization is interacting with the environment when there is cold in the early stage of the plant. So something like that, parameters like that are usually modeled, and they are different from individual to individual, but they have really low interaction with the environment. But we know how they interact. So they don't change their values from environment to environment, or have very low change from environment to environment, but we know how they interact with the environment. And why we have to integrate both models together? So crop growth model usually is able to predict new environments, but it failed to predict new genotypes. And to have a good modeling through crop growth model, what you need to do is for each individual, it run one individual by individual. So you cannot extend to new individuals, but for one cultivar, and usually we do crop growth modeling for the major commercial cultivars that have very high or large data for a single cultivar, and we try to estimate the GSPs for them. And crop genome prediction, what is good at predicting new genotypes, but it has low accuracy in predicting new environments. And the thinking was, when originally combining both of them, is that if we combine both of them, what we are going to have is we are able to predict new genotypes in new environments. And this was the original thinking of the model. And actually, the other advantage that is why that model is frontiers is that it transfers crop growth model from being a cultivar-based approach to a population-based approach. And this is the main advantage that we are getting here. And this is why it's improved the accuracy for predicting new environment. But in our research, during the past 3 years at AVR, we found that there are even more that we can get, or more things that we can get from integrating both models together, and these are listed here. So crop growth model, as I said, they require really large data of for each cultivar in across wide range of environments to have accurate prediction of the GSPs. But with CGM-WGB, we found that we need less data compared to that. And we found also that detecting QTL affecting specific physiological process is possible. So if we run GWAS on the GSP, it's possible to detect QTL affecting different physiological processes. And it's also performed better in predicting in unrelated individuals. And we were also able to predict some traits where we don't have even phenotype for them, and I will explain that later. And even if you have missing environmental variables, we were able to predict these missing environmental variables because crop growth model usually very demanding in type of the number of input data that we need, and some of the data may not be accessible, especially for historical data, but we find that we can approximate some of these environmental variables. So when we combine both of them, which is, if you don't have them, crop growth model will never work. But with CGM-WGB, you still have a good accuracy and you still can predict these missing environmental variables. So I will go through these. So what we use is, when we combine with genomic prediction, we combine a wheat growth model, which has 13 genotype specific parameters or latent traits. And this model, it just take the different modules, which include the phenology, the crop biomass, and the crop nitrogen. And from the environment, it also modeled the management, the environmental factors, and the soil water. And just to talk about how accurate the first advantage that we get is to get more accurate results from CGM-WGB compared to CGM with small data sets. So we have a reference data that is in one environment also only. And this is usually not a valid data for crop growth model. And we try to estimate the GSPs on this data. And we try to estimate the heritability of the GSPs. With CGM, you can see that it's only on average for all of them, it's 7%. While with CGM-WGB, it was 0.81, which is almost 11 or 12 times higher with the accuracy. And of course, when you have more heritable traits, when you apply GWAS on them, you can get better results because they have better estimation and more accurate estimations for them. And when we did GWAS, whether it's CGM or CGM-WGB, for CGM, we were not able to detect any QTL, but for CGM-WGB, we detect different QTLs. And you can see for phenology, for example, we detect the major photoperiod and vernalization genes for wheat, which are very important. And we detect some really well-known QTL for yield, for example, and for protein, controlling the protein. And yeah, and this QTL has really well demonstrated in literatures that are affecting something similar to these traits. I will not go through that. And the third application was the ability to predict unrelated individuals. So what I did here is dividing the population into three subpopulations based on their distribution of the PC. And these are individual in gray here. I just marked them to remove them, just because they are a hybrid between the populations. So what I did is to use one population to predict the other two populations. And you can see that for traits like grain yield and grain numbers, they got really much higher improved accuracy with CGM-WGB compared to the standard genomic prediction models. So, and this is not surprising because, as I said, when you use the GSPs or the genotype specific parameters, they are purely heritable traits. So when they are purely heritable, when you go from individual and trying to predict them in a new individual, because they have high heritability, they are more accurate. It's more likely to get them accurate in new individuals, even if or in unrelated individuals. It's more likely so for highly heritable trait to get them more accurate on these individuals. The other one is the ability to predict trait that even if you don't have them in the reference. So for our analysis, what we have, the trait that we measured are heading date, maturity date, protein content, grain number, and yield. So what the thinking was, because now we are not depending on the phenotype of these traits directly to to calculate the accuracy for them. We are depending on the GSPs and how the GSPs are interacting to produce the final traits, which are these are the final traits. So can we, if we estimated these GSPs and assume that I don't have a measure for yield, or I haven't measured yield, can I predict yield through GSPs without having yield in the reference to calibrate the CGMWGB model? And for the phenology-related trait, the heading and maturity dates, we found that heading date, we were not able to predict it very accurately when we masked it. So what we did here is just we assume that we don't have a measure for a single phenotype for this trait, for heading date, for example, and trying to predict it through the GSPs. And the same for maturity date. So we found when we masked that, we masked maturity date, we were able to accurately predict it, and the accuracy did not get affected. But that wasn't the case for heading date, which is predictable, because heading date is usually something in the middle of the phenology. So if we need a trait that in the middle of the plant life, just to calibrate different phenotypic stages, while maturity is at the end of the simulation. So the end of the simulation is easier to be modeled. But if you don't have a point in the middle to be to calibrate the models, such as the heading date, if you didn't include that in the reference, so you cannot have an estimation, a good prediction for it. So, but something we can do is to use other phenological stages, such as putting or something else, so which can be easily predicted with the image analysis. But that's something we are currently working on. The other thing is protein and the nitrogen. And just here, I want to point something at the WGP perform much better than whether the CGM-WGB, whether we're in masking trait or not for the protein. And it's mainly because the protein module in our crop model was not that very good module. So you can see even when the protein data is included in the reference, the prediction accuracy is lower than the standard genomic prediction model. And when we even masked it, we were not able to predict it accurately. And this is also predictable, because the nitrogen module, if you look at the nitrogen module, we have only a single trait for it, which is protein content. And when we mask this one, we have nothing to calibrate that module. So we found that for each module of the crop growth model, we need at least one trait to be able to predict it accurately. The other thing, when we trying to mask grain number and yield, when we try to mask one of them, so the other one can accurately predict it through the CGM, and the CGM-WGB will not have any accuracy difference between whether when the trait is masked or not. But something you may have noted that still for grain yield and grain number, the WGP model or the standard genomic prediction model perform much better than the CGM-WGB if you find here. So it's 60%, 0.6 the accuracy, but here it's 0.53 for yield, for example. And when we try to investigate that later, what we have to do is that we found that CGM is very sensitive to the input data. So for example, in our analysis, and this is what direct me to the last benefit of CGM-WGB, is the ability to approximate missing environmental data. So in our analysis, what we did in the previous analysis, in the previous slide, we don't have, there is a very important parameter, which is the initial soil water content. So we assume that the initial soil water content is equivalent to 0.35 for all environments, because we didn't recorded that. And this, if you back to the previous slide, you find this is the reason. We find that this is the reason, because we don't have an estimation for initial water content. This is the reason why CGM-WGB performed less than WGP for the biomass module. And what we did after that, we tried to, as we sample GSPs, we tried also to sample the missing environmental variant, which is initial soil water content. And you see here that the CGM here is the original analysis from the previous slide. It's lower than the accuracy here, but the accuracy has been increased, and with the with sampling. So when we try to sample them like we sample the GSPs, or not the same way, in the similar way, you can see that the prediction accuracy become just equivalent to the WGP. And why we didn't get an improved prediction accuracy is, it's again because other people who use the same model, got an improved prediction accuracy when they try to predict a new environment. But unfortunately, this was one of the limitations in our data, that we have data from a single location. And the current research that we are doing now is trying to have more environmental data across different environments and trying to predict the accuracy across different environments. But this is still too early to mention that. And finally, I just want to finish on, and it's good that John described the breeder equation at the start. Just want to back to the breeder's equation and to show how each or how each component can be improved, or how to improve genetic gains through each component of the breeder's equation. So the first thing, or the intensity of selection, we have to carefully select that to avoid what we call running out of heterozygosity or running out of diversity, which will limit the additive effects. So the second thing, we need to always continue to increase the genetic diversity and adopt new resources, for example, exotic germ plants in plants, which usually has high unexplored genetic variation. For the accuracy, we need to try to apply new novel models such as the CGMWGP. We need to increase our reference population. We need to use the model, or trying to get different data sources such as environmental management, high throughput phenotyping, omics, whatever data you can have access to, trying to include them, trying to find a statistical way to include them. We need to model genotype by environment interaction well, and maybe also we can trying to have non-additive effects such as the dominant effect or abscesses effect. And we need to reduce the length of genomic of the breeding cycle, and that by using genomic selection at seeding stage, for example, or using double haploid or speed breeding. And one of the main things that genomic prediction is, many reports have found that genomic prediction may not be more accurate sometimes, in some cases, or in many cases, wasn't more accurate than the phenotypic selection. But the main advantage that we get is reducing the cycle time of the breeding cycle. So instead of having, for example, 10 years of breeding cycle to develop one cultivar, it's reduced to 5 years. And this was the main advantage of genomic selection, rather than having a more accurate selection. But in some cases, of course, especially when you use advanced model, you can even have higher prediction accuracy. And finally, we need to simulate different, just to evaluate that on their effect on genetic gain, different components. We need to have always simulate different breeding schemes to assess the potential to improve genetic gain. And in conclusion, so genomic prediction or genomic selection holds lots of potential, and there's still very big room for innovation and to adapt new method and new models. And of course, CGMWGP seems to be one of the most promising models that can be used, and as it can see that more heritable and less genetically correlated GSPs, and we can select on specific mechanisms or specific physiological mechanisms using GSPs, and we are able to predict unrelated individuals in untested environments, and even for traits that have no phenotypes. And even if we have missing environmental variables, we can predict those, and it gives us a new insight into different physiological mechanisms, things that the gene that we detected with GWAS on the GSPs, for example. And what is for the future, what we do, we need to exploit more sources of data and to integrate more models together to get different source of information or different source of data types. And still machine learning hasn't been really well exploited, and we need to think of different way to integrate machine learning within this approach and CGMWGP in the future, because it doesn't, for example, need, as we proved, that it doesn't need a phenotypic for some traits, something like yield. So can we use it other traits, something like the image analysis that we can get from image analysis, for example, to calibrate the model without having to really measure any of the complex traits or the time-consuming traits, of the laborious traits. This is something that was investigated in the future. And also, can we use greenhouse data also to calibrate or to predict field data? This is something still an open question. We should work on it. And thank you. So, and I hope that getting your questions now.
Thank you very much, Abdul. That was a great presentation. Really enjoyed seeing how crop growth models can be incorporated into genomic selection. So we do have a few questions and have a reasonable amount of time to get to them. So I'll just tee them up. The first one's not addressed to anyone in particular, so perhaps one or both of you can comment on it. The question is, are there traits which are more amenable to genomic selection? And how many traits can be stacked in genomic selection approaches? Is there a limitation? And perhaps you could speak to the prioritization of traits work on in genomic selection.
Okay, so do you want to answer, Michelle, or I can start?
I can start, and then you can maybe fill in with a bit
Okay.
more detail.
Yeah.
So just as an example, for instance, in ryegrass, so there are absolutely traits that are more amenable to selection. So typically, the higher perishability, the easier it is to get a high prediction accuracy. A number of traits can be stacked. I would say there probably is going to be a limit. You're going to have just simple biology is going to interfere. So, for instance, if you have two traits of interest that are negatively correlated, so as you increase one decreases, but you want to increase both, that's going to be more difficult to select for both of them together. So you do need to sometimes do a bit of exploratory work with your phenotype data to figure that out. In ryegrass, to prioritize traits, we do use a selection index. So particularly we look at yield at seasonal time points. So we prioritize different seasonal yield. For instance, ryegrass does very well with spring yield. So we don't really want to focus on improving spring yield, but we do want to bring it up, for instance, in autumn and summer. So we give a little bit more weight to our selection index to that trait to prioritize individuals with better performing, say, autumn yield. And we don't do so much focus to spring yield, but we don't ignore it either. So a selection index is how you will help prioritize your traits, and you can develop that on a number of different parameters, such as
Yeah. So from a statistical point of view, you can run it trait independently, or you can run multiple trait models. But for most application, we currently use a single trait and approach. But there are no, we can work on any trait without limitation. But we usually prioritize the trait that the industry are the most important to the industry. So that's how we prioritize our traits. So the most commercially important traits will come first. And yeah, so yeah, and main trait in stacked and approach there. Yeah. So the limitation usually for low heritability traits, generally, which are more harder to be predicted, but usually we solve that by increasing the reference population.
Great. So another question is asking about, can you please give some details about the actual population sizes in your ryegrass reference population, training both genotyped and phenotyped, as well as the candidates for selection, the ones that are tested and genotyped only. Any suggestions regarding the optimal population sizes for a complex quantitative trait like FHB?
Yeah, so in terms of the ryegrass reference population, we have a couple of different ones. So the one example I talked you through from a breeding program for biomass yield, I think that was between 800 and 900 cultivars, or part of our reference population for that. So they're both phenotyped and genotyped. And then in terms of selection candidates, that could vary. So this program was run across a number of years, and every year this breeding company will send us their selection candidates. And that could be anywhere between 1,000 and 2,000 plants, really depending on budget for sequencing and how many plants they have grown on the site. As Abdul mentioned, if you have a low heritability trait and you have a complex trait, a bigger reference population is going to be better. But there's no one number that's going to fit every species and every trait. Bigger is usually better.
Yeah, for the optimal population size, usually there are some equations that based on your trait, its heritability and your population, your effective population size, you can try to give a prediction of what is the optimal size for your population, that in theory, but the best thing is to try, if you have site trying to because at some stage you reach a stage where you cannot get a higher prediction accuracy, whatever you increase the size of population. So the best way is to test that, but there are some theoretical equations to approximate that.
Okay, thank you. Another question, can you comment on the roles of the classical GS genomic selection models built from the infinitesimal model proposed by Fisher and machine learning, deep learning models based on prediction work models in future GS work? And secondly, how can we apply genomic selection to speed up the genetic gain, but at the same time preserve genetic variations in a breeding population?
Okay, for the first one, while GBlob is kindly, or you can get it as in somehow it's in an interphysimal model, because you assume that it's limited, of course, by the number of the analysis is limited by the number of SNPs you have, but it's in somehow it's the interface for the interphysimal model. We can do it with GBlob, and currently with machine and deep learning, we still really don't have the right population to make advantage of machine learning and deep learning in the future, because still our population are few thousands. We did not really expanded that. We are currently in the animal, we have a population for one trait, which is over a million individual. And we are hoping that this will have will show an advantage for machine learning, but so far nobody has ever showed any advantage when using machine learning or deep learning over the standard models. And this could be mainly due to the limitation of the populations we have, but in the future we may have better models. And it's also worth to investigate if we are able to integrate different data types within a single deep learning model. So that's something, but still it's still, it's very this type of research still at very early stage. So hopefully in the future can work, but not yet there. We are not yet there. And how we can speed up genetic biology? Well, I think maybe Michelle can comment on the second, or maybe better than me. Yeah.
So I guess in terms of speeding up genetic gain, primarily the way that genomic selection does that is to shorten your selection interval. So both John and Abdul showed that previous equation. So you can increase your genetic gain by increasing the selection intensity, increasing the selection accuracy, for instance. And you can do both genetic selection, you can have that as well, but primarily it's going to be by shortening that generation interval. You can select a particular plant when it's just a seedling before you're able to measure any sort of meaningful traits on it. So that's the primary way. And in terms of preserving genetic variation in the breeding program, so the method that we have personally used for ryegrass is to apply a fitness matrix between all the potential candidates to ensure that they're not too closely related. Things like that. There's a number of different approaches. I don't think there's any one set way that people will use, but just to make sure that you're not only selecting very closely related individuals, you can look at the allele diversity of your selection set and make sure that meets the threshold, for instance. So there are a number of ways. It is very, very important to do that. And I think another way as well that's becoming a bit more common is to model or simulate the population variance of your selections and to make sure that it's actually quite broad, so they have future variation selection.
Yeah, from statistical point of view, what we usually do is to take the relatedness among individuals and we treat it in somehow like a trait, and we try to reduce that relatedness in the selection candidates or the selection on the selected population, the output.
And here's a question about the cost of genotyping. So this person is asking about how much it costs to do the genotyping platform that Michelle presented. They've come across some papers saying you can reduce the set of markers to pretty small levels, say 100, to reduce the costs and maintaining prediction accuracy. Maybe you could comment on that, and could you share any insights you have in the context of genomic selection in ryegrass?
Yeah, so firstly, I don't have an exact dollar amount in my head, but for the target assay we've developed, it is sitting under $50, I believe now for sample. In terms of using, say, 100 markers for genomic selection, this is really going to be strongly dependent on what species you're working in and even what subset of population you're working in. So if you have a highly inbred species, it's very homozygous that you're working with, and there's not a lot of variation, and you're basically just trying to capture recombination events, and you don't need a ton of information, then yes, you could probably get away with something like 100 markers. Maybe you could, maybe you have a very good set reference set for imputation, and you can keep that up. In terms of ryegrass, typically what we work with in the plants that we're selecting among, there's too much diversity, and the LD breaks down too quickly to use 100 markers. For us, I don't think that's something that I don't think we would reasonably be able to get down to that level and maintain a prediction accuracy with that the current resources and with the current level of diversity that we work with. As I mentioned, though, we are looking to design a lower density target capture assay, and maybe we won't be able to use that in the novel material, but as I said, if we have, for instance, sequenced parents to high density, look at offspring, we have some information from the parental generation that we can use to help us impute up from lower density to high density. So yes, it can be done, but again, it's going to be really dependent on what you're working with. You're probably looking at more inbred or very closely related individuals to develop. So typically, genomic selection accuracy increases as you increase the number of markers to a point, so it does plateau. But I think you're in a very specific set of circumstances if you're going to be able to use just 100 markers.
If you want to get more rough estimation about the number of markers you need, there is some statistics called effective number of segments or effective number of LD blocks. An estimation depend on the effective population size and the LD in the population and the genome size. So there are some equations that can do that. It gives you a rough estimation how many markers, But of course, the number of markers is not enough, the distribution across the genome. So theoretically, what you need is within each LD block, you need at least one marker to represent it.
And Uzal Litton online has a few questions that he wanted to ask. Maybe we could go to Uzal. You still on?
Yeah, hello. Can you hear me okay?
Yeah.
Yes, we can hear you.
Okay, so my first question is like in terms of like the diversity in phenotype, like what do you think? Like the high diversified population is good for the training population? What is your comments on that?
Well, yes. Yeah, usually, yes, the variation. We usually want to improve that, but at the same time, we need to have you have to make balance. So yes, you need larger variation of the phenotype and larger variation in the population, but you need also to assess your marker density. Everything needs to be assessed. Each component that is required for the estimation, the SNP effects should be assessed well to cover that. So if you want to increase the genetic diversity, you need to increase the that's your marker density as well. So yeah, but yes, definitely increasing the phenotypic variation is important.
Yeah, like why I am asking this question, because I'm using the GBS SNPs, and it's quite like high diversified SNPs, and I'm using this one to predict the DON content in the population, and like when I use like the RR blob model, the phenotypic correlation, like the true phenotype and the estimated phenotype correlation is, I'm getting a maximum of 20%. So that is quite low to predict a genotype. So do
Yes,
you
but
have any
what is
suggestion?
what is the heritability of your trait?
Okay, I didn't check the heritability in statistically, but it's that one is not highly heritable, but it's a medium type of heritable trait. It is.
Yeah, generally this is one of the reasons. If you have low heritability, another reason could be if you have really low genotyping density, but you still have very high diversity, so you are not capturing all that. So yeah, one thing you can do is to estimate the heritability, the SNP heritability, and comparing to the actual heritability, if you estimated that. So by SNP heritability, there are different tools. The simplest one is, what was the name? MTG2, I guess, and GCTA, I guess. Two software can do something like that. It estimate the SNP heritability. And if it's low, it's fair, and you have to check for heritabilities on the same, or in a population with the same level of diversity as yours and the same size. So you have to check if they are getting the same level of accuracy. But just what to do is something need to be really well considered across different factors. What is your what is your SNP density? What is your effective population size? Different factors should be taken into account before you know how to increase the your prediction accuracy. What is your population size? The reference population size is also important. So there are different factors that would affect that. It's not, a straightforward answer. You need to assess everything in your population to know what you have to do exactly to increase the population size, or to increase the prediction accuracy.
Okay, yeah, that's a good answer. Thank you for that one. And like, do you have anything, any comments on the different types of models that you already presented the SNP metrics with the different types of models, but do you think, like, is there anything that we could work on the models as well?
Definitely yes. Yeah, the problem is that most of the best models are not really available to everyone. So for example, CGMWGP is not really available to everyone to apply. And it's really complex, not easy to be applied. But for the other models, the Bayesian models, there are BJLR, for example. You can just try different models through that and try to compare the accuracy for your traits. Maybe you are using gblob while you have some genes that with a really high effect that are not really very well captured because the distribution is not, or the distribution of the SNP effects is not, doesn't fit your trait. So you could check with the models. You could get few, some models could work much better for your trait, for your specific trait, but other factors need to be considered. So for example, what is your population size, the reference population size?
So the reference population size is 300.
300. And how much you are using for training?
For training, normally I use 80 to 90 populations.
Oh, that's really small. That's very, very small. Yeah. So that might be one of the main reasons. What I would recommend in your case to you, because it's even your population is 300 is small.
Yeah.
If I will try to do that, I will try to use 200, for example, and 50 for the training and 50 for validation, but I will do lots of replications for that analysis. If you have good computational resources, you can have large number of replications just to make a good estimation of the accuracy.
Okay, so like your suggestion is to make the replication of the population and their phenotype as well?
No, not replication. You need to increase your population size.
Okay,
and if you are
got it.
If you are using just only 100 individual for training, that's really, really small number. And for me, that's the main reason why you are getting 0.2.
Okay, got it. Got it.
Yeah.
Yeah.
So
Thank you.
try 250 and 50 for validation, but
Yeah.
you need to have lots of repeat the same analysis with different set of validation. So just randomly assign your population into reference and validation, and the 50 for validation will need to be changed every time. So each replication should have different 50 lines for validations. Okay. If you repeated that process, if you have good computational resources, if you repeated that for 100 times of random splitting and repeating the estimation of the accuracy, then you will get roughly a good estimation with fairly good standard error or standard deviation for the estimation.
Okay.
But
yeah,
you need to
thank you.
increase the reference population size. That's the comment number one I will have to you.
Yeah, thank you. Some other questions are already have been answered. Thank you for that.
Yeah, you're welcome.
So I'd like to thank Michelle and Abdul again for your presentations. It was great to see them. This is exciting and an important field. And thank you for everyone for participating. Accelerating breeding is an important suite of technologies and strategies that will be key to helping us meet our challenges in plant breeding and for today and tomorrow. If the presentations spurred a question or an idea, don't hesitate to contact us at partnerwithus@gifs.ca. And we'll welcome any conversation. If we are unable to assist, we'll put you in touch with somebody who might be able to. Thank you very much.
Thank you.